So today we are going to know about the delta notch signaling pathway. It is important in cell-to-cell communication system and it is found mostly in the multicellular organism. It is important in embryonic development, cell fate determination and neurogenesis and stem cell maintenance. So there are Notch receptor. These are the transmembrane protein present on the surface of the receiving cell. Delta or Jagged. They are the transmembrane ligand present on the surface of the signaling, that is the neighboring cell. Then ADAM protease and gamma secretase. Enzyme that cleave the Notch receptor, they are the ADMA protease or gamma secretase. Then Notch Intracellular Domain. Activated part of Notch that enter the nucleus, that is that Notch Intracellular Domain. Then steps in the delta notch signaling pathway. There is a ligand-receptor interaction. The delta on one cell bind to the notch on the neighboring cell, and this interaction it occurs within the this juxtacrine signaling, this adjacent cells, and there is the binding. This binding trigger the two sequential cleavage of notch. So it is known as the proteolytic cleavage. First by ADAM protease, that is extracellular, and second by secretase within the membrane. Then NICD translocation. The Notch intracellular domain is released and it is translocated to the nucleus of the receiving cell. This NICD bind to the TSL, that is CBF1 suppressor of hair length or lag1 transcription factor. It converts CSL from a repressor to an activator. The targeted gene like His and He are activated and this regulate the cell fate decision. So what is a fate? Fate is a final outcome of what the cell will become. It is a biological destiny, the shape by the signal and interaction like delta notch. Here, for example, if there is a tissue, then it will form the same tissue or a different tissue, that is its fate of that particular cell. So some cell have a fate to become a nerve cell, other have a fate to become a skin, blood or gut. Means these cells will convert into a skin or a blood cells or gut cells. So they are changing its fate to become a particular cell in developmental embryo. Means formation of hands or formation of legs, all this is the fate of that particular cell. Now signaling from other cell like delta notch, gene expression and the location of the embryo is important. So cell fate is what the cell is becoming. Cell specification is cell is starting to commit but still flexible, and cell determination is cell is locked into its further fate. That means once the cell is determined, it cannot change its fate. Once our hands are become, then it cannot be converted into legs or something else. And the cell specification is that stage where the cell can determine whether it has to become a hand or whether the embryo will form a leg or anything else. So that is the cell specification, and cell determination is the final stage. Now it cannot be converted back into any other organs. So differentiation is the cell actually becoming its final type. Visible changes, all the changes are seen in differentiation. Now fate is the final outcome of what a cell will become. It is a biological destiny, the shape by signaling and interaction like delta notch. So the key function of delta notch signaling are the processes and the role. For example, in later inhibition, the role is to ensure the neighboring cell adopt different fate. Example, one become a neuron and other do not. Then in stem cell maintenance, the delta notch helps in maintaining the balance between the self-renewal and differentiation. And the process of angiogenesis, it regulates the blood vessel branching. And in organogenesis, it guides forming a tissue like pancreas, heart and nervous system. So it has some clinical and research relevance. For cancer, aberrant notch signaling is implicated in T-cell leukemia and breast cancer, etc. and congenital disorder. For example, mutations in the Notch component can cause various syndromes. Regenerative medicines, in that also it is critical and important for cell stem based therapies. Important example of juxtacrine signaling in animal development. It allows adjacent cells to influence each other's fate without release of a diffusible signal. The ligand and the receptor are transmembrane protein ensuring strict spiral control of signaling transmission. Now we know that what is a notch? A single pass transmembrane receptor on the responding cell. Then delta is a transmembrane ligand. It is found on the surface of the signaling cell. Then these are the transcription factors CSL. The nuclear effector in the receiving cells are CBF1, SUH, LAG1. Then NICD is a Notch Intracellular Domain that activates the fragment that enter the nucleus. Then signaling mechanism. We have known that the ligand binding takes place, then sequential proteolytic cleavage takes place, and NICD release and nuclear translocations take place, and then final transcription activation. So these are the steps of this delta notch signaling. Let's revise. When a delta expression cell make contact with a notch expressing cell, the extracellular domain of delta bind to the extracellular domain of the notch, and this interaction induces some conformational changes in the notch. Then sequential proteolytic cleavage, this conforms changes making notch susceptible to proteolytic cleavage. First, ADIM family metalloprotease cleave the extracellular domain near the membrane, and then gamma secretase, a multi-protein complex also implicated in Alzheimer's disease, cleave the transmembrane segment with the lipid bilayer. Then Notch intracellular domain is released from the membrane and rapidly translocated in the nucleus of the receiving cell. Then transcription activation is done in the nucleus, and ICD binds to the DNA binding protein CSL, converting it from a repressor to a transcriptional activator, and this complex recruits co-activators such as the mastermind to drive expression of the targeted genes such as the HES and HE transcriptional repressor that inhibit the pre-neural gene expression. One of the most studied role of Notch signaling is lateral inhibition during the neurogenesis. In a group of equivalent cells, one that express more delta activation, more notch in its neighboring, suppressing their ability to become the neurons. So this ensures only one cell become a neuron while its neighboring take an alternative fate. For example, glial cells. And this interaction exemplifies a negative feedback loop where the initial difference in delta expression is amplified to produce the distinct cell fate from the homogeneous population. So there is some developmental importance and pathogenic implications of this Notch signaling pathways. So in developmental importance, it helps in stem cell niche, blood vessel branching, neurogenesis. In pathological, it helps in oncogenesis, developing disorders. Notch signaling pathway is remarkable in that it allowed a cell to control the fate of its immediate neighboring through a simple mechanism that combined precise contact, limiting proteolysis, and direct transcriptional regulation. And this mechanism exemplifies how evolution has optimized signaling system for efficiency, fidelity, and spatial specificity in multicellular organisms. Thanks for watching. Do like, share, and subscribe.